Nick Oswald, welcoming you to this bite-sized bio web seminar. Today's presentation is titled qPCR Assay Design 101, and it's being presented by Lou Lee. Lou is a research and development scientist at Biosearch Technologies, where he specializes in qPCR applications, and I for one am really looking forward to benefiting from some of his wisdom on this topic. Today's webinar is sponsored by Biosearch Technologies. You can find them at www.biosearchtech.com. Go check them out after the webinar and see what solutions they have for your research. During the presentation, please type any questions that you have into the questions box on the right-hand side of your screen, and we'll put them to Lou at the end. So Lou, now over to you for your presentation.
Hi Nick, thank you for the introduction, and thank you everyone for chiming into the webinar. As Nick mentioned, if you have any questions, feel free to chime in at any point during the webinar, and I'll try to field as many as I can at the end. Otherwise, if you can see on the main screen here, there's an email, techsupport@biosearchtech.com, and I could always answer these offline for you. So today, we're going to go over the design of primers and probes for quantitative PCR. And a little bit of an introduction, the invention of polymerase chain reaction, or as we like to call it, traditional PCR, has allowed us to replicate and quantify DNA. But it wasn't until the discovery of quantitative PCR, or qPCR, how we've been able to measure expression with precision and sensitivity. So what we have illustrated here on the left is a traditional PCR image. What it is, it's a post-PCR read on an agarose gel, where you're taking your PCR product and running it out against a known ladder. So you can see there's a little bit of a guessing game to predict the size of these bands. On the right is an illustration of a qPCR assay. There's two methods of detection here. One is a cyber assay, which uses a DNA binding dye for their detection. The other is a probe-based assay that uses a fluorescent oligo. For the sake of this webinar, we're just going to focus on a probe-based assay. So what is qPCR? It's an accurate and precise way of measuring the quantity of the target in a sample without having to open the reaction tube for subsequent analysis. It is done by monitoring the kinetics of a signal generation, as opposed to traditional PCR, which only reports on the endpoint product following amplification. So on the right here, this is an illustration of an amplification plot for quantitative PCR. As we mentioned, traditional PCR is a post-PCR read, so you're measuring this in this plateau phase, whereas quantitative PCR, you're measuring in the exponential phase. So to elaborate a little bit more on this, during the exponential amplification phase, the sequence of the DNA target doubles every cycle. So essentially, every cycle is a twofold change. However, as the reaction progresses, some of the reagents are going to be consumed as a result of the amplification. This depletion will occur at different rates for each replicate. The reaction is going to start to slow down, and the PCR product is no longer going to double at each cycle. So this is represented in this linear phase. Eventually, you're going to reach a point where you're going to get complete depletion. The reaction is going to slow down, if not stop altogether, and that's going to be this plateau phase. So as we mentioned in the previous slides, where traditional PCR you're measuring in this plateau phase, you could guess that you might see some variability, which is illustrated here, where we have 96 replicates of the same sample, and in that plateau phase, you definitely can get variation, whereas qPCR measured in the exponential phase, you're still seeing that high level of precision. So what to take out of that is that accurate quantification is the defining feature when you're comparing qPCR versus traditional PCR. So here you can see the different applications that you can use with qPCR. Absolute quantification incorporates the use of a standard curve, which we'll go into in a second. Relative quantification uses a comparative cycle threshold method without a standard curve. Plus-minus assays is mainly used for high-throughput screening. A lot of people like to use cyber in these applications. SNP genotyping. SNP stands for single nucleotide polymorphism, which is essentially two alleles that differ by a single base, and we'll also get into that. So as mentioned with absolute quantification, it incorporates the use of a standard curve. So the advantages of using a standard curve is that you have the ability to accurately quantify your signal. This is done so by taking a template with a known quantity amount and doing a serial dilution. So this example here uses a 10-fold serial dilution. So as we mentioned, every cycle has a doubling effect. So in theory, for a 10-fold dilution, you should see a 3.32 cycle difference. If you want to do some quick math, if you take 2 to the power of 3.32, that's going to equal 10. So with qPCR, it allows you to set a threshold, which you see as this red line, which is going to allow you to associate a cycle number to a fluorescent value, which you could plot out on a graph here that's going to generate a slope value that you can use to calculate PCR efficiency. So if you plug in this minus 3.32 value here, it's going to come out to 1, which equals 100% efficiency. So as we mentioned, a 10-fold serial dilution, in theory, should give you a 3.32 cycle difference, and you'll notice with the slope, it's minus 3.32. So going over the concept of qPCR, as mentioned, we're just going to focus on a probe-based assay. Instead of explaining it, I'm going to show you a little illustration that we have set up that you could find on our website. So what exactly is a BHQ probe? Essentially, it's a linear oligo that has a 5' reporter dye and a 3' BHQ quencher. BHQ stands for black hole quencher. It's a non-fluorescent quencher. And so you'll see what's illustrated here is the motions of a PCR, where you get the denaturation of a double-stranded DNA into single strand that allows for the anneal and extension of the primers and probes. So I'm going to stop this here and go back to the presentation. So you'll see it illustrated here at the bottom. So as the polymerase extends during the process of PCR, your probe is going to get cleaved. And this cleavage is going to release the reporter dye from the quencher. So this is how you produce your signal that you can measure. But while the probe is still intact, it's getting quenched in a couple different manners. One is called static quenching. So static quenching forms a ground state complex of the probe through direct contact, direct physical contact. You can see that illustrated there. Unlike static quenching, FRET, which stands for forced-or resonance energy transfer, it's a through-space mechanism in which energy from the reporter dye is transferred to the quencher without absorption or emission of light. So essentially, in this graph here, you'll see that the absorption spectrum of the acceptor must overlap with the emission spectrum of the donor. So the key thing to take out of this is FRET quenching is extremely distance dependent. So it's very important when you design a probe that it has a certain length. Normally, the cutoff with FRET, where it starts to rapidly diminish, is 30 bases. So with that being said, with the nature of the business of qPCR, you're going to run into a lot of probe types. We're not going to be able to cover them all. I'm just going to mention a few here. We here at BioSearch offer 2 different probe types, BHQ probes and BHQ plus probes. I'm going to mention NGB, which stands for minor group binder probes, only because a lot of times it's associated with our probe types. But one thing you'll notice is that each one has a different range as far as their length. So when you're looking at desired traits in the design, you want to probe sufficiently long enough to ensure binding at the proper temperature, while sufficiently short to ensure quenching efficiency. So it's kind of important to find a happy medium as far as length and the melting temperature of the probe. So for certain applications such as gene expression, we generally recommend you start out as a BHQ probe design. You may have certain instances where you're not going to be able to generate a design, and more than likely it's due to a problematic target sequence, such as something that's AT-rich. In these instances, we recommend that you switch over to a BHQ plus design. So the reasoning behind this is that BHQ plus probes allow you to design shorter sequences with relatively high melting temperatures. This is due to the duplex stabilizing technology that allows for the shorter sequences. It's due to modified bases that we incorporate that boost the TM. So with those properties of a BHQ Plus probe, we also only recommend them for SNP genotyping designs because, as some of you know, SNP genotyping, the designs must be specific enough to discriminate the mismatch bases. So when comparing MGD and BHQ Plus probes, you have to keep in mind that the design, they are each designed according to separate proprietary chemistries, so that each changes the thermodynamics of probe binding. So these different probe types cannot be converted without adjusting their sequence design to compensate for the change in binding properties. So what does that mean? It basically means that you can't always transition directly from MGD to BHQ Plus probes. So with that all being said, these probes need to be designed using a software that incorporates their unique contributions. So we here at Biosearch, we've come up with our own software called Real-Time Design. It's basically going to help you facilitate the design of BHQ and BHQ Plus probes for qPCR and SNP genotyping. So I'm going to leave this here and go actually into a live demonstration of our software. So Real-Time Design is actually an internet-based software. There's nothing to download. It's free to use. All you need is a login. So you can go to our website, biosearchtech.com, and log in if you have an account. Otherwise, you can create an account for free. And I'll just log into my account here. And you'll see there's a quick link right here to Real-Time Design on the main page. So clicking on that will take you to an introductory page, which will go over the different features that are offered on the website. Once you're done with that, you can click Dual Labeled Probes to go into the main portal of the software. So you see it's very simplified, but there's definitely a lot of information that can be found using the carets and the drop-downs here. So one of them is a way that's going to show you how to input your target sequence. So this can be done in 3 different ways. One, you can copy and paste in your sequence. Another is you can enter in the NCBI accession number. And the third method is with this drop-down, you could upload if you have a saved sequence. So I actually have a saved sequence here. We'll just do the copy and paste method into the software. There's other features down that I could show you as well. You have the different applications. So we kind of went over this with gene expression. You could either have a BHQ or BHQ Plus design, and also SNP genotyping, which is strictly a BHQ Plus probe assay type. Also with the different mode, you could do either custom or express. Batch mode allows you to enter up to 10 sequences at the same time so that you don't have to manually do that. We'll go into include, exclude a little bit later, but the different species type you could enter in. I believe this sequence being used is mouse, so we could select that, input the sequence, and hit next. So this next page is going to allow you to set different parameter sets. Normally with express mode, there's nothing really to touch here. You could also select a different probe type if you need. For now, we'll just leave that as default, and you would just hit next. So what the software is going to do is, as you saw on the last page, it has 3 different parameter sets. It's going to go through the most restrictive parameter set first to try to design the primers, primer set, and probe. If it doesn't meet the criteria there, it's going to move down to the next level and then keep going until it finds a design. So we'll just let this go through the motions and see what turns up. Okay, and so from here you see what the software does in express mode is it gives you just a single primer probe assay design. And if you need to get additional information, you could click on this checkbox right here. So it'll list out the forward and reverse primers as well as the probe sequence. It's going to give you a little bit of information there as far as some of the parameter settings. And at the bottom, you'll see this qPCR submission. So clicking on this, what it's going to do is I'll let this run in the background, but what it's going to do is it's going to evaluate the forward and reverse primer to see whether or not they're specific to a single target, a single variant, et cetera. So this is going to give you your specificity of your design. Other features that you could see here is if you click on the assay itself, it'll illustrate the primers and probes where they are on the actual target sequence. And at the bottom here, you can see them listed out again. So we could see if that search is done. We'll reload this. But essentially, as mentioned, what you ideally want to see is a single ePCR hit. So with this sequence, you notice there are 2 results. So this could be a number of things. It could be a splice variant. It could be 2 sequences that share similarities. But just know that these assays are not specific. They essentially could be amplifying at 2 different regions from the genome. So what can you do to fix this? So if you go back at your original target sequence, you could blast this. So NCBI has a tool called BLAST, which I pulled up here, but there's the link. BLAST stands for Basic or Local Alignment Search Tool. And so what you could do is you could review this sequence and blast it. And essentially what the software is going to do, this tool, is it's going to look for repeat regions within the sequence. And so this shouldn't take too long to run. I was sort of worried with this since this is a government website and with the furlough and everything that there might have been delays, but I think everything's going to run smoothly here. Fingers crossed. If not, I have an illustration that I could show you instead. Sorry about that. But essentially when you do the alignment, the blast search, you'll see there might be some gray areas. So this represents a repeat sequence. And so if you notice in the design, this is around base 60 to 80. But the RTD primary probe design, lo and behold, you'll see your amplicon is in that region. So that's probably why we got 2 ePCR hits. So what you could do is you could mask this region. You could do so by just placing a series of ends. Normally put about 6, so that the software is going to know to exclude this region. So we go back. I've already gone ahead and done this here. We could take this edited sequence and plug this in. Actually, it did turn up. So there you go. You can see that. Same thing illustrated. There's a few repeat regions there. So you would just edit those out. By going back into the software, we could go back into the portal and re-import this with that edited sequence and just let it run through again. I'm just going to take some parameter sets away just to speed it along. And if we go through and we rerun the ePCR, we'll see if it cooperates with us this time. If not, I have it illustrated here. So it's going to result in a single ePCR hit. So this is ideally what you're going to want to see. So once you have, oh, I believe it's finished. So there you go. So there's a single ePCR hit. So once you're satisfied with the design, you could either export it into an Excel file and save it for later. You could, if you're logged into your account, which you should be, you could add it to your cart if you're ready to order it. Also, if you're for some reason unhappy with this design process, you can go back to the design module. So as I mentioned earlier, there may be certain instances where you designed a BHQ gene expression assay and you didn't come up with any results. So let me illustrate an example there. So although this says BHQ+, we're going to design this as a BHQ probe. It's basically going to fail. Just keep in mind, it's not failing because the software is failing. It's failing because there's an issue with the target sequence itself. In this case, it's an AT-rich region. So the failure is going to come from sequence. But this is just a good example to see what you would expect when you come up with 0 results. There's different workarounds. So as you can see here, no assays were designed for this. So what you could do is click on the diagnostic, and this will tell you where it failed in the design process. So you see it's complete for the forward and reverse primer and the primer set, and it failed in the probe design. So you could expand this out to see where the failure was, and you see there's a high failure count because the TM is too low. And so we had mentioned previously the difference between BHQ and BHQ plus is there's modified bases that boost the TM. So what we would do in this case, we could go back and we could switch this to a BHQ plus design and paste in that same sequence and just let it run through. So you definitely do have options if you run into issues doing the design with express mode. And there you go. So you see that we get results here, and you see it's BHQ plus now. And you can see that illustrated on the target sequence. And you could export and add to cart at your leisure. So another example I can go through is if you have, say you have two different sequences and you know that there's a lot of similarities between the two sequences, but you want unique specific designs for each one so that they're not going to detect each other. So with this, you can interrogate the two sequences by using an alignment tool. And I have it illustrated here. I'll also have it later on. I can provide it to whoever needs it. But you could take the two sequences that I've just called variant A and variant B, and you can copy them and paste them into a multiple alignment tool. This one in particular is just called Clustyle W2. And you would just hit submit. So what the software is going to do is, as it stated, it's going to align the two sequences, look for homology between the two, and then give you results. So as you can see where all the stars are, this means that they're exactly identical. So you'll see, if you scroll down, it's pretty identical to each other. So what you want to do is you want to find a region between these two that differ. So lo and behold, you have this small section here. And so what you want to do is you want to truncate the rest of this down to just this area. So I've gone ahead and done that at the bottom here, and I just called them variant A and variant B edited. So I just included this small region. And let me expand this out. And also what I've done is there's this little character, it's called a tilde. And so what this does is it triggers real-time design to just focus on this area as far as the amplicon design. So the amplicon is up to about 200 bases. So it's going to try to flank this region in the design. So you could plug these two sequences into real-time design, and in theory, your design should be discriminatory towards each other. Just for the sake of time, I'm not going to go through that. So our next example is a SNP genotyping assay. And so with SNP genotyping, you can enter in the sequence three different ways. You can go to this link here that goes to a SNP database, and you can obtain the DB SNP ID, which is this RS number, or you could actually copy and paste in the actual sequence itself. This one has an IUPAC code, and this IUPAC code here can be represented at the single base mismatch. Or you could just enter in the actual bracketed SNP itself. So we'll just take this one and use that as an example and go back into real-time design and make sure that you are doing a SNP genotyping assay. And we could plug this in. And you'll see here that now it has the options for the two different alleles that you have. You could change the dye labels if need be. Otherwise, you could just let the software go through express mode and do the designs. But going back to here, as that runs, essentially what you're going to expect to see when you do allele discrimination, this is a post-PCR endpoint read where you're comparing allele 1 versus allele 2. So each one has a different fluorescent label. So you expect to see some clustering. So if it's homozygous for one allele, it'll cluster in this fashion or for the other one. Or if it's heterozygous for both, it'll cluster towards the middle. And you can see the representation here, which I could also show you if this is complete, which it is. So you see the two different BHQ plus probe designs with a single base mismatch and you can see it illustrated on the target sequence right there. So I'll go back to the beginning here, and I'll just use this sequence as an example. But suppose that whether you saw it through a publication or you had your own designs, but you had like a primer pair, and you only wanted the software to design just the probe. So you can use this function, which is called include/exclude oligos. And when you enter in your sequence and you check that box, so when you go to the next screen, now you'll see an area where you could anchor in those oligos. You could actually exclude those oligos if needed. I do have a primer here, and I'll enter in the reverse primer. So the software is going to evaluate these, and you'll see that they might come up with some errors through the most restrictive parameter set, so we could loosen that up a bit and go to the least restrictive, and you see everything's green, everything looks okay. And when you hit next and go through the parameter sets, the software is only going to design a BHQ probe. And you'll see here that's the same primers, and you just get the probe. So you can see that there's a lot of features within real-time design software to allow you to make designs for quantitative PCR, SNP genotyping, and so it's a very useful tool. And with this, I'm just going to go to any sort of questions that we might have.
Okay, thank you very much, Lou. That was a really great presentation. Those the BHQ probes in the real-time design software look like a great package. They make qPCR assay design a lot easier and more flexible. I think you did a great job of demonstrating that there. We have some questions coming up. Just a note to everyone they can, if you have questions off the back of this, you can still add your questions into the questions box. So the first one is, can you expand further on how the SNP genotyping assay you showed, how that works?
So essentially with SNP genotyping, as I mentioned, it's only a single nucleotide base mismatch. So what you have to do is you need to design two separate probes with two different fluorescent dye labels so that you could differentiate between the two.
Yeah. Okay.
If we go back to the website here.
I think that was mainly what this is about getting the animation up again. It looked like a great explanation, but just to run through it again.
Yeah, if you go to our website, you go under support and then downloads.
Yeah.
At the very bottom, that illustration I showed you earlier, it's located here. It's called Probe Formats Animation, and that's going to open in the background. And you can go through BHQ Plus probes and look at that illustration.
Yeah.
But yeah, if you have additional information, I might not be able to field it here, but for sure, and use our tech support email that I'll show you after this animation. Sure. But you see that with this illustration, the BHQ Plus probes are shorter in sequence, and it has the modified bases to still boost the TM. So you see those modified bases are called propine counterparts or pyrimidines.
Okay. And the basis of the assays that you use two probes, is that correct?
Yeah, so you have to, since you have two alleles, you have to differentiate them somehow, so you have the two different probes.
Yes.
With two different dye labels.
Okay. And then they outcompete the one that you generate a signal from the one with the correct match, and that's how you distinguish which, as you're showing there.
Yeah, exactly. And then you do a post-PCR read, which is the allelic discrimination that does the cluster plot. So it should, in theory, cluster towards whether it's homozygous for one allele, homozygous for the other allele, or heterozygous for both.
Okay.
You could always confirm that if you have positive controls for each allele that you could run.
Yep. Okay. I think that as well, the answer is to point people towards that as a really useful section of your website in those demonstration animations.
Yeah, so you can see that with the mismatch, it should only anneal to one or the other according to that mismatch.
Yep. And it's the activity of the TAC on the hybridized probe that is releasing the signal. Yep.
Yeah, so once the TAC extends through the sequence, it's going to cleave the probe and release the reporter from the quencher to give you your signal.
Yeah. Okay.
Okay, so there's that.
Cool. All right, so a question here from Chow asking, can you explain, again, internal quenching, the concept of that?
Okay, I didn't really go into too much. I was trying to more emphasize on how important the length of a probe is, and normally our cutoff for a standard BHQ probe is 30 bases. There might be some instances where you might have to have a longer probe sequence. So what we do recommend there is we have internal quenchers. What you could do is if you have an internal T base, you could append the internal BHQ quencher. You just want to make sure it's about 10 bases or more from your floor, your reporter die. You also need to ensure, since now on the 3' end you don't have a quencher, you have a spacer of some sort of that's going to block the PCR extension.
Yeah.
So that's a possibility to have an internal quencher for longer probe types.
Yeah. Okay. And the next question from Frank. Can probe-based assays use melt curve analysis to verify the specificity of the product, or is subsequent sequencing required?
I would say yes and no. I don't think it's the most efficient method. That kind of goes back to the other detection method for qPCR called cyber, where cyber, it's incorporating just a binding chemistry. But the issue with cyber chemistry is it's going to detect anything double-stranded. So then you sort of want to run that melting curve analysis post-PCR to just make sure you have a specific product by just looking at the peak that's going to correspond to a certain melting temperature. So in using melt curve analysis with probe chemistry, I don't see it too often.
So you mentioned that BHQ plus probes. Aside from SNP genotyping, what other situations are those recommended in? As
I recommend, if you're doing gene expression and you try to design it as just a regular BHQ probe and you get no results. So it might be a situation where you have a problematic sequence, such as like an AT-rich region. So the problem there is that since it's so AT-rich, the probe length is going to be way too long and it's going to be outside of the parameters. So with the benefits of a BHQ plus probe design, it allows for a shorter sequence in these AT-rich regions because you have modified bases that boost the TM. So you have short sequence with still relatively high TM to make for a design.
And is the difference between the normal BHQ probes and the BHQ plus probes that's the different chemistry, the different
yeah,
bases.
It's those modified bases that are appended to them.
Awesome. I'm just checking to see if there's any more questions to ask. We'll keep it to this last question here from Frank. How many BHQ or BHQ plus reactions can be multiplexed in one tube?
That's actually a good question. You might have certain limitations. One is going to be the multiplex itself. The other is going to be the instrument you're using. So certain instruments don't have the spectrum to differentiate between different probe types. We've seen certain instruments you can multiplex up to 5 dyes as long as they have 5 different filters or 5 different settings to differentiate between each dye label. So we do offer a wide variety of fluorescent dye options so that you can multiplex. The other inherent issue that you might run into with multiplexing is there's a wide level of competition associated with multiplexing. For the most part, for new users, we don't recommend you multiplex more than 2 at a time. For advanced users that are familiar with primer probe optimization and changing different aspects of their protocol and their assay, it's definitely possible. You know, we've tested in-house up to 5 dyes before.
I guess the more complicated you make it, the more area there is for problems.
Yeah, if you want, I could bring up, we have something called a dye selection chart.
Mm-hmm.
So if anyone can see on the screen, we have a wide variety. All the ones with the little icon next to it is what we offer. those are proprietary through our company. Everything in bold is what we also offer, and you can see we have different replacements. If you look at the second page, this is a multiplexing recommendation, so it shows all the different instruments that we've reviewed, and you can see some of them have 4-plex or 5-plex options.
Okay, that's an excellent resource. We'll post a link to that in the
also,
the
if
video
you want,
page.
under support, under applications, there's a multiplexing little overview here that you can go through, and you can see there's that multiplexing chart.
All right, even better, yeah.
Yeah, so we can provide these links for whoever needs them later on.
Yeah, okay, and we'll add them alongside the video on the website. Cool, okay. So I think that brings us to the end of the questions and the end of the seminar, so thank you again, Lou, for an excellent presentation.
Yeah, thank you for hosting us.
Cool, no worries. And also, again, I'll thank our Biosearch Technologies for sponsoring this webinar and encourage you to take a look at their website. You can see that they have a lot of resources in there that would benefit you. Last, but by no means least, a big thank you to our audience for taking the time to attend today. If we didn't get to your question, we will try and answer it for you offline. If you've enjoyed the seminar and you'd like to view the video recording of the session, please visit the seminars page on bitesizebio.com, and it should be available there within the next 24 hours. And there you can also check out our upcoming webinars. So, until next time, good luck in your research, and goodbye from all of us at Bitesize Bio.